Thank you so much for joining us on this video session of the mSystems Early Career Special Edition presented by Flore. Was there a moment as you've advanced your career where things started to connect and where you realized you were working on something that could affect human health?
Really, one of the things that really stuck with me was this shift in understanding that I always thought about these microbes operating independently. And then by starting to doing this research, it really became obvious that just like when we take a pill by mouth, it can affect everything in our body. The same goes for our microbes. Anything that they produce in our body makes it into the portal blood system and can affect things from cognition all the way to allergies. And so all these processes are also interconnected. And so that was very much the aha moment for me, kind of realizing, as naive as it is, how interconnected we are with our microbiota.
In your mind, what was the aha moment for you where you began to read or understand that the gut microbiome is going to have a key part in human health in the future?
Well, I think for me a really important changing moment was during my PhD early on. I was comparing the gut microbiome of guinea pigs to that of humans. And superficially, on a very high taxonomic level, they actually looked very, very similar. But the more fine-grained you went, the more you went from species and maybe down to strain level, the more difference there were. At strain level, there was basically no difference at all, no similarity at all between human and guinea pig. And this was for me the moment that probably our human gut microbiome is extremely well adapted or specifically adapted to us as humans. And it's the same for the guinea pig and probably the same for the dog and the cat, etc. And this was for me the moment where I thought, like, okay, if it's so well adapted to us, then probably there's also an important health aspect to this, right? So that they can provide very specific benefits to just humans that they cannot provide to a cat. Or a cat might need some very specific other benefits that it gets due to its cat gut microbiome.
Help us understand what drove you to enter this field to begin with. What piqued your interest as it relates to the microbiome?
I started my research into the microbiome actually not from a human angle, but from a microbial ecology angle in desert soils. And it was quite interesting because I did a postdoc at the University of Pretoria in South Africa, where my group was investigating how microbes contribute to soil health. And as part of that, I went to the Namib Desert. And my first investigation that I did was into the microbial community, and specifically the viral community of a hypolith. And that's a rock, a type of rock microbial community. So imagine you're in a desert and there's barely anything growing. And there's this white rock lying there, you pick it up, and on the other underside, you have this microbial community that's green and it's dominated by cyanobacteria and it's actually like alive among what seems to be nothing.
Understanding of human health and human gut microbiota, what have you been learning that can make a crossover into the interplay in humans?
Yeah, that's a great question. I think there's a lot of potential for crossovers, and really it's understanding how microbes interact with all of these environments. There are such similarities in terms of that. There's a limited operating space that the microbes work within. For example, if we think about the different biogeochemical processes, when these are taking place within the human microbiome within the human body, they're not so different from how they're taking place in any of these other environments. And so the human body is a wonderful model ecosystem or environment to understand how these processes are taking place, understand how dynamics and survival and succession are taking place. And it gives us such an insight when we go to these other environments and we try to understand what's taking place, what's happened in the past that has given rise to the microbial community that we observe at the moment that we are visiting that environment, because of course we don't have the historical context that we often do when we're studying the human microbiome. So it's a great way to understand the fundamental processes that lead to an emergent microbial community at the time that you visit.
What was that aha moment for you where you realized there's an impact to human health related to this type of work?
Right. Well, So one of my projects I was working on was really a more mechanistic, kind of fundamental study of trying to see what the role of virulent phages were in the gut microbiome. And at the time, it was kind of hard to study because there's so many different species that are present there. It's hard to track all of these individual species. And so we took a more reductionist approach, which was to use gnotobiotic mice, which are essentially germ-free mice that are colonized by a defined community of microbes, colonize them with a subset of bacteria, introduced virulent phage, and used molecular methods like qPCR and next generation sequencing to track each member of that community. What we found was that introducing phage could knock down the targeted bacteria, 1 out of those 10 species. But that's not where the effect stopped. Really, the phage and the bacteria coexist, and it had a resulting effect on the co-colonizing species. So, those species that interact through interbacterial interactions, those interactions were modulated based off of the targeting of different bacterial species in there.
Is that sort of a long-term evolution study, or are you looking at specifically between a parent and a child and how much is preserved, whether in an insect model or a human model?
I think my point is that, lots of animals inherit microorganisms from their parents, and that's a very different evolutionary relationship than microbes that we might acquire horizontally from the environment. And so, a better understanding of the evolutionary biology of inherited microorganisms is relevant to broad classes of microbes that are transmitted vertically from parents to offspring rather than horizontally transmitted between individuals or things that we just pick up from the environment. And so we're trying to understand the evolution or the coevolution that occurs between an insect and a single microbe, just as a way of better understanding the processes that shape heritable microorganisms and their hosts.
I don't typically see the word holobiont in my reading. Maybe start there and help me understand, what is a holobiont?
So holobiont is essentially a relationship between the host and its entire microbiome that is either living inside it or around it. So you can think of the host and its associated microbiome as an ecological unit, one unit. So that unit is defined as a holobiont.
How will we keep both gut microbiomes healthy?
One way is certainly to now just map everything that we can find and try to understand, if we look at plants that are healthy and have a good yield, can we find some markers there that differentiate them from those that behave less healthy or are less healthy.
As well, when I was in my Stanford days, I was lucky enough to be with David Crive and David Relman looking at one of the very first studies that looked at viruses or phage in the human mouth. So again, as a kid growing up, it's been, looking at microbes always around us, and that, I've always wanted to do this, right? I've always known that to be a thing, right? I think the newest thing now is how many are beneficial.
So if you had to go back and redo some of those experiments, what type of technology might you use today?
Oh, well, that's a great question. I think today when we're sequencing is so much more available to us, we can really try to understand kind of a systems level analysis of diseases. So we were really focused, because I came from an infectious diseases laboratory, you were focused on just the pathogen itself. So we were screening for genetic variations within the pathogen that might explain the variation in disease outcome. But there's definitely variation also that's related to the human host. And then there's the variation of the microbiome. And nowadays you can really profile both host, microbiome, and pathogen at the same time to really understand this kind of systems level interaction. That's definitely what we're trying to do now in my new lab at Johns Hopkins.